Okay, let's unpack this next stage. We've talked about grouping linked markers.
Right, figuring out which ones tend to stick together.
Exactly. So today, the mission is the next big steps. Actually ordering those linked markers along the chromosome.
Putting them in their proper sequence.
And then turning those genetic relationships into real map distances, centimorgans.
We're looking at the methods, the algorithms, the math behind building those detailed genetic maps.
It's like going from just knowing towns are connected to having the actual road map, showing every single intersection and the distance.
Precisely. And it's fundamental. You need this map before you can even think about finding genes for traits, like in QTL mapping.
So, okay, we've got our linked group. How do we figure out the order? What's the basic principle?
Well, the core idea is pretty straightforward, actually. Markers that are physically closer on the chromosome are just less likely to have a crossover event happen between them.
Less recombinate.
Exactly. So ordering is all about finding the arrangement, the sequence, that requires the fewest implied crossovers overall, minimizing recombination events across the whole set.
Okay.
For just 3 markers, let's call them A, B, and C, you can just compare the recombination frequencies between each pair.
Like AB, AC, BC.
Yep. If, say, the recombination between A and B and between B and C is lower than between A and C,
then B must be in the middle.
B is most likely between A and C. Simple enough for 3.
Right, but I'm guessing it gets way more complicated with, hundreds or thousands of markers.
Oh, absolutely. That's where it gets really complex. It turns into what's called a combinatorial optimization problem. It's famously similar to the traveling salesman problem.
Finding the shortest route through many cities. I've heard of that.
Exactly. Trying every single possible order for even a modest number of markers, it's computationally impossible. The number of orders just explodes.
How impossible are we talking?
Well, the sources mention that for just 20 markers, trying every order brute force could literally take centuries.
Centuries.
Yeah.
For 20. Okay. Yeah. Algorithms are definitely needed then. So what kind of clever tricks do scientists use?
Right. So early methods tried to build a map sequentially, add markers sort of one by one or step by step.
Okay.
Algorithms like branch and bound were smarter than brute force. They'd add markers based on some criteria, trying to limit the possibilities they explored, like minimizing the sum of adjacent recombination fractions. SARF.
SARF. Keeping local crossovers low as you build.
Kind of, yeah. Or rapid chain delineation, which starts with the tightest linked pair and builds out. But even these struggle with the massive data sets we get now.
So for the, millions of markers from modern tech, what's the approach?
That's where multidimensional scaling, or MDS algorithms, really shine.
MDS, how does that work?
Think of it like this. You have all these pairwise relationships, recombination frequencies, kind of like a messy web connecting all the markers. MDS tries to represent those relationships in lower dimensions, like 2D or 3D space. And ideally, the markers that are truly neighbors on the chromosome will fall along a nice line or curve in that lower dimensional space. It reveals the underlying order.
So it helps you see the structure hidden in all that pairwise data.
Exactly. It's powerful for ordering thousands, even millions of markers. It can also help spot problem markers or even structural things like inversions if you compare it to a reference map.
And when you have different possible orders from these methods, how do you pick the best one?
Generally, likelihood is considered the gold standard criterion.
Likelihood, why that?
Because it directly calculates the probability of observing all your actual data genotypes, missing data, everything given a specific proposed order and map distances. It's a robust statistical way to compare different map possibilities and find the single most probable one.
Okay, so algorithms like MDS find likely orders, and likelihood helps pick the winner. But you mentioned earlier that the initial recombination frequencies themselves might be a bit off because of noisy data.
That's right. Missing genotypes, maybe dominant markers where you can't tell heterozygotes from one homozygote, general uncertainty. It can bias those initial pairwise estimates.
So how do we refine them once we have the order?
That's the job of multipoint analysis. Instead of just looking marker to marker, it uses all the markers in their determined order simultaneously to get better estimates.
And the key tool for that is?
The Hidden Markov Model, or HMM.
HMMs, right. What makes them so good for this?
They're perfectly suited because genetics on a chromosome fits the model's assumptions. You have hidden states, the true, unobserved sequence of genotypes along the chromosome.
It's really there.
Exactly. And then you have the observed states, the actual genotype data you collected, which might be incomplete or noisy.
Your measurements.
Right. HMMs use probabilities to figure out the most likely sequence of hidden states, true genotypes, given your observed data. They rely on conditional dependency, meaning the probability of the true genotype at one marker depends only on the true genotype state at the previous marker in the sequence, which makes total sense for markers lined up on a chromosome.
Okay, that connection makes sense.
So using the order, the model considers transition probabilities, the chance of a recombination between markers, and emission probabilities, how likely your observed data is given a hidden true state. It uses all the data together to refine those recombination frequency estimates.
So it cleans up the noise by looking at the whole picture, the context of the ordered markers.
Precisely. Our sources showed an example where initial estimates, like say 0.22 and 0.37, were refined by the HMM to maybe 0.25 and 0.33, leveraging information from all markers. It gives you more accurate recombination fractions.
Got it. More robust estimates. Okay, final step then. We have the order, we have refined recombination frequencies. How do we get to the actual map distances in centimorgans, the units people use?
For that, you need a map function.
Why can't we just use the recombination frequencies directly?
Because recombination frequencies aren't additive. Genetic map distances measured in centimorgans are supposed to be additive. You know, the distance from A to C should be A to B plus B to C if B is in the middle.
But recombination frequencies don't work like that. Why not?
Because of something called interference. One crossover event can actually make another crossover less likely to happen nearby. It interferes.
Oh, okay.
So a recombination frequency of 1.4 isn't necessarily twice the genetic distance as a frequency of 0.2. Map functions correct for this interference. They translate the non-additive frequency into an additive map distance.
Yeah, are there different functions?
Yes. Simple ones like the Morgan function assume no interference, which usually isn't realistic. The Haldane function is another. But the Kosambi function is very commonly used.
Why Kosambi?
Because it models interference in a way that seems to fit a lot of real biological data better. It accounts for the fact that interference levels can change depending on how close markers are. Applying Kosambi, or a similar function, converts those refined frequencies into additive centimorgan distances.
Right, making the map distances behave like actual distances you can add up.
Exactly. That gives you the final usable genetic map.
So quite a journey then. From grouping linked markers, using algorithms like MDS and criteria like likelihood to order them, refining the numbers with HMMs, and finally using a map function like Kosambi to get additive distances.
That's the process. It turns that raw linkage information into a structured quantitative map of the chromosome.
It really is building a detailed blueprint, which makes you wonder, with these powerful algorithms and models getting so good at handling noisy data, refining estimates, even spotting structural changes, how might they further change how we understand genetic variation, especially its impact on traits across diverse populations or changing environments?
That's a really interesting question to ponder, isn't it? How much deeper can these maps take us?